So what are we talking about today? Today in this lesson we're going to focus on what are viruses, how are viruses structured and what type of viruses exist in the world, and how are they really different. How does a virus utilize the lytic and lysogenic cycles of viral reproduction? How does the DNA of a virus relate to DNA of its host? Because interestingly enough, a big part of us is viral DNA. And what are some examples of common viruses? So if we're looking at this, here are a couple of viruses that you might see. Measles, West Nile virus, even the common cold are all caused by viruses. Okay, and the first question that we really want to ask is, are viruses living? And viruses are not living. They're not living for a couple of reasons. First, they don't respire, right? They're not releasing energy. Okay, they are also not sensitive. They don't express sensitivity, right? They don't respond to the environment. Okay, and they also don't really grow and reproduce, right? So they're not growing and reproducing. They are therefore not living, right? And here you can see, here is a cell being infected by a number of viral particles, and these viral particles are not living. These are composed primarily of a protein coat, and then there's DNA or RNA, some sort of genetic material inside them. So how are viruses discovered? As you're looking at this, right, this is a tobacco leaf, and it's mottled kind of in color. So you can see that the bottom part of the leaf kind of has a discoloration, and that's due to the fact that this tobacco leaf is infected by the tobacco mosaic virus. Okay, so what was first done, right? So if we just back up for a second, right? So here's the tobacco leaf, right, with the tobacco mosaic virus, and what happened initially? They went ahead and they extracted the sap from these diseased leaves and then put that sap onto leaves that were non-diseased. And they found that that would go ahead and cause the non-diseased leaves to become diseased, right? So that there was this transfer, okay? And they said, all right, well, that must be caused by bacteria, because we know that bacteria might carry this information, but they cannot visualize or find any sort of bacteria under the light microscope. So at this point they're saying, well, okay, maybe it's just bacteria that's small enough that we can't see them. And that actually proved not to be true. And so what they went ahead and did was this, right? So they extracted the sap. They passed the sap now through a filter that actually is able to filter out the bacteria. And so they now know that when they rub it on the leaf, that there's no bacteria present in that sap anymore, and it's still causing these healthy plants to become infected. So there's actually some sort of pathogen, right, that it isn't bacteria, right, that's existing within it. And that led them to go ahead and hypothesize that, okay, there's actually something smaller, much more simple than a bacteria that's probably causing this, and they figured out at that point that, okay, it is leading to what we now know as viruses, right? And this is what the tobacco mosaic virus looks like. It's kind of rod-shaped in this way. It actually uses RNA as its genetic material instead of DNA, right? And you can see that here as well, right? So you've got this protein coat, this capsid, if you will, right, with the RNA kind of inside it, right? And so this is where we're really first kind of starting to discover what viruses are at this point, right? So here's a couple of different viruses. So we can see the tobacco mosaic virus, we can see the adenovirus, and we can see influenza, right, which is causing the cold. And so if you notice, right, there's very different structures that we're seeing here, right? So we're seeing some viruses that are made up of RNA, some that are made up of DNA, right? You're seeing on the surface of some of these glycoproteins, right? If you're looking at influenza, right, it actually has a membrane. And viruses, right, are really simple, okay? Extremely simple and extremely small, right? I mean, the smallest viruses are something like 20 nanometers in size. So you could go ahead and fit millions of these on a pinhead, right, if you will. And those viruses are smaller even than of a ribosome that's within a cell. Okay, and we also know too that the smallest viruses maybe have something like 4 genes within them, right? And the largest ones maybe have like several hundred genes. Whereas bacteria in comparison are having maybe 200 to a couple thousand genes. So viruses are very simple, right? Even though there's kind of this diversity of structure that exists kind of between them. So here, right, this is an example of the adenovirus, right? And if you're looking at the adenovirus here, this, right, so this part here, this is the genetic information. And in this case, the adenovirus is carrying DNA as its genetic information, right? You can also see, right, it has this capsid, right? And this capsid is this protein coat, right, that's kind of surrounding it, right? We've seen this before when we're talking about like the T2 phage, right, which is made up of primarily either genetic information and then also protein. And then you can see these glycoproteins, right, that are also kind of surrounding it. And those are going to be involved kind of in attachment with the virus to its host cell. So over here, we also have the influenza virus, and you can see here it's using RNA as its genetic material. So sometimes we're using RNA, sometimes we're using DNA. If you notice too, the influenza virus actually has a membrane around it. Okay, so it has a membrane surrounding it, and oftentimes that's really useful for allowing it to infect its host cell if its host cell also has a membrane. Okay, and here again you have these glycoproteins, and those glycoproteins are going to help with again recognition of proteins that are on the surface of the host cell that it's going to infect, and it's going to allow it to go ahead and kind of enter the cell. So how do viruses reproduce? That's really the question that we want to ask ourselves now. And as you can see here, right, so here's the virus, and we have the virus, right, so here's that capsid, which is that protein coat surrounding the genetic information, and let's say in this case it's a DNA-based virus, right, and so it's going to enter the host cell. And so oftentimes you have what's called like a lock and key fit, where, as we talked about, they have glycoproteins often on the surface of these viruses, and that's often going to interact with a protein that's on the surface of the host cell, which is the recognition between those two is going to allow this virus to go ahead and enter, right? So as that virus enters the host cell, you can see that the protein from the capsid goes ahead and breaks down, right? And then here's the genetic information. So here's the DNA. Now, a virus, right, requires a host cell. Really, it's really an intracellular parasite. Right, because it can't exist on its own. And what it really needs is it really needs the machinery of the cell to make copies of itself, right? Because viruses, they lack ribosomes and really the other machinery to make DNA copies, and also protein. So essentially, in essence, right, the virus is going to go ahead and hijack this cell. And again, it depends upon its host range in terms of what it's able to affect. Right? I mean, for example, measles is only able to affect humans, right? Some viruses have a greater range of species that they're able to infect, right? And some can only infect one species. Also, I mean, some viruses target only specific cells within a eukaryotic organism. For example, AIDS, right? I mean, AIDS only affects certain like white blood cells, right? That exist within a eukaryote. So what we're seeing here is the DNA, right? The DNA is going to get copied. So basically, the virus is hijacking the machinery of the cell to make copies of its own DNA. And it needs these new copies of the DNA to go ahead and build more viruses, right? That's its goal. It's also going to make mRNA from that DNA, right? So it's going to do transcription. And then it's going to go ahead and it's going to do translation. And so as it does translation, it's going to make more protein for its capsid, right? So what you're going to see is that protein from the capsid and then the DNA, that genetic information, are going to assemble right into new virus. And as you have these new viruses, they're going to go ahead and they're going to exit the cell, right? And as they exit the cell, they're often going to go ahead and burst the cell, right? Because they don't care if they destroy the cell. That doesn't really matter to them. Their goal is to hijack the machinery of the cell, go ahead and use it to make many copies of itself, and then it can go out and infect new cells.
A virus is an intracellular parasite that can reproduce virus consists of a nucleic acid genome enclosed in a protein shell called a capsid. In the virus shown here, the genome consists of DNA, but some viruses have RNA. Some viruses are also covered by a membranous envelope that is derived from the membrane of the host cell. There is usually a lock and key fit between the proteins of the capsid and receptors on a particular type of host cell. The virus attaches to a host cell, and viral DNA enters the cell. The viral DNA uses nucleotides and enzymes of the host cell to replicate itself. The viral DNA then commandeers other host cell materials and machinery to transcribe its genes into messenger RNA and translate the RNA message into capsid proteins. Viral DNA and capsid proteins then assemble into new viruses. Mature viruses leave the host cell, often destroying the cell in the process. The viruses can go on to infect other cells, spreading the viral infection.
So now that we've discussed the viral reproduction cycle in some kind of as a general concept, and you've also seen an animation of it, we're going to look at the Lytic cycle and the Lysogenic cycle in a little bit more detail and kind of focus on how a phage might use this to go ahead and infect a host cell. So here's the Lytic cycle, right? And so the Lytic cycle, the first thing that's going to happen is, so you can see here's the phage, right? So the phage is going to go ahead and attach to the cell. Okay, so when it attaches, it's then going to go ahead and you're going to see that the DNA from the phage is going to enter the cell, right? And the protein coat, the capsid, is going to go ahead and remain outside, right? Then what's going to happen is it's going to go ahead and it's going to use the machinery of the cell, right, to go ahead and make multiple copies of its genome and also its proteins. Okay, so again, it's going to go ahead and do replication, it's going to use that, and it's also going to go ahead and do transcription and then translation, so it can go ahead and make proteins. What's ultimately going to happen is that's going to get assembled, right? Because when you're looking at the phage, it's made of this protein coat, right? It's made up of actually multiple different proteins. You've got the head, you've got the tail, and the fibers, which are all made out of protein. And then you have that DNA, the genetic information that is stored within the phage, right? So all of that's going to go ahead and assemble after that. Okay. What's going to happen then is once there's enough phages that have been built at that point, they're going to go ahead and burst the cell, right? So they're going to be released. And at this point, probably about 100 to 200 of those phages would then be released from the cell. Okay. So if you remember, bacteriophages are a virus that are specifically infecting bacteria, right? So it's going to go ahead and release those 100, 200, if you will, phages back out, and then those will go ahead and they can infect new bacterial cells. Okay, so now at this point, right, let's go ahead and take a look at an animation.
During early infection, the virus attaches to the bacterium. Then the virus injects its DNA into the bacterium. The viral DNA uses cellular machinery to reproduce itself. During late infection, components of the viral protein coat are produced from genes in the viral DNA. The viral particles self-assemble into new viruses. These viruses overrun the host cell and burst free, lysing and killing the cell.
Some phages also go ahead and use what we call the lysogenic cycle, right? And so if you noticed in the lytic cycle is going to go ahead and kill the host cell. And here, as you can see in the lysogenic cycle, right, that's a little bit different, right? And so the lysogenic cycle, I want you to focus down here, right? So what happens is the DNA from the phage actually go ahead and integrates into the host cell's DNA. And then it becomes what we call a prophage at this point. So this is going to allow, right, it's the DNA from the phage has become a part of the host cell's DNA. And so then every time that the host cell copies itself, right, then it's going to create a new copy of itself. And that new copy of the bacterial cell is going to include the DNA from the phage, right? So it's going to go ahead and create multiple copies of itself, right? So it's going to copy the DNA of the phage, right, because it's included within it multiple times, right? So you can see that here, okay? So, but there are also times, right, so that's what's happening in the lysogenic cycle, able to make multiple copies of this DNA without killing the host cell. But as you can see here, sometimes, right, we have phages that are what we call temperate phages, and they're able to go ahead and switch between the lytic and lysogenic cycles depending upon the environmental conditions. And a good example of that would be like the common cold sore, right? It goes ahead and it copies itself, it's not virulent, right? It's not causing any harm. And then when the environment changes, it actually switches into the lytic cycle and causes cells to burst, right? Which is what we were talking about previously. So those are called temperate phages. They're able to switch between both the lysogenic and lytic cycles depending upon what the environmental conditions are around it.
In contrast to the lytic cycle, the lysogenic cycle reproduces the viral genetic material without destroying the host. The lysogenic cycle of phage lambda begins when a phage binds to the surface of a host bacterium. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The viral DNA is incorporated into the host cell DNA, where it is called a prophage. Every time the host bacterium reproduces, it replicates the phage DNA along with its own and passes the copies on to daughter cells. Occasionally, the phage DNA exits the bacterial chromosome and initiates a lytic cycle. The viral DNA takes over the metabolic machinery of the host cell to make phage DNA and proteins. The host cell lyses, releasing phages which can infect other cells. This diagram summarizes the lysogenic and lytic cycles of phage lambda.
Most of the viruses that we talked about up to this point are DNA-based viruses, but that's not true for all viruses, right? Some can use RNA as their genetic material, and we're going to take a particularly close look here at HIV, which is standing for human immunodeficiency virus, right? So it's going to attack the immune system, which leads to the onset of AIDS, right? Which is the acquired immune deficiency syndrome. So what you're seeing here is HIV, right? HIV has this capsid, this protein coat, right, surrounding the viral envelope, and then you have RNA in it as well as reverse transcriptase, right? And so seeing this ACE part, you know that this is an enzyme. And so since this is an enzyme, what it's actually going to allow the virus to do, it's going to be able to turn RNA into DNA. And so we can see this here, right? So what you're seeing, and it also, if you notice, it has these glycoproteins on the surface. So what you're seeing, again, like via lock and key fit, it's going to go ahead and its glycoproteins are going to attach probably to proteins that are on the surface of that host cell, and then it's going to go ahead and go into the cell, right? And so you can see here is the RNA, and then also this reverse transcriptase. And what reverse transcriptase is going to do is it's going to allow it to take the RNA and make a DNA strand from that, right? And so it's going to do that actually twice, and then it's going to end up with its double-stranded DNA. And then what actually happens is this DNA gets inserted into the DNA of the host cell, okay? So it's going to be inserted into the nucleus. So now this DNA is in the host cell, and at this point, right, what we call this is the provirus, or the proviral DNA. And so you have this proviral DNA that's inserted within here, right? So this is DNA. And what's actually very interesting about this is this DNA will remain incorporated in the host cell. It will never leave the host cell, right? Whereas we were seeing within kind of bacteriophage and the lytic cycle, that actually leaves the cell. But this DNA remains within the cell. And so what it actually is going to do is it's going to take the machinery, right? And it's going to do transcription so that it ends up with the mRNA that it needs, okay? And then what's going to happen is, right, you're going to have the RNA that it wants, and it's going to go ahead and be able to assemble that, right? It's also going to take the RNA, and it's going to be able to make the reverse transcriptase, right, because that's going to be a protein, right? So it's going to do translation. And it's also going to go ahead and make these glycoproteins. So really interestingly, right, it's taking this RNA-based virus and it's able to make the RNA, it's able to make the reverse transcriptase, it's also able to make these glycoproteins, and then it can assemble all of that back into making the new HIV virus, right? And the key thing again is that that DNA is remaining within that cell, so then it's able to go ahead and use that machinery again, right, and create more copies of itself. So this is a really interesting way in terms of how this RNA-based virus is working.
Understanding the structure cycle of HIV, the AIDS virus, is critically important to dealing with the disease. HIV is covered by an envelope derived from a host cell membrane. Glycoproteins studying the envelope recognize and bind to receptor molecules on the host cell. A protein coat surrounds the viral genetic material, which consists of two molecules of single-stranded RNA. The enzyme reverse transcriptase enables the virus to make DNA from an RNA template, a trick for which this group of viruses is named retroviruses. The RNA molecules of HIV enter a host cell when the virus fuses with the plasma membrane and the coat proteins are removed by enzymes. Reverse transcriptase catalyzes the synthesis of a DNA strand complementary to the viral RNA strand, and then a second DNA strand complementary to the first. The double-stranded DNA is incorporated as a provirus into the host cell's chromosomal DNA, where it may lie dormant for years. Occasionally, the provirus is transcribed into RNA. This RNA serves as both messenger RNA for the formation of HIV proteins and as genetic material for the next generation of viruses. Protein coats form around viral RNA and reverse transcriptase molecules. Viruses bud from the host cell, acquiring envelopes as they leave.
So how do we go ahead and treat viruses? One way that we're going to do that is using vaccines. So you're going to go ahead and use a harmless variant, right, or a derivative that's going to go ahead and stimulate the immune response, right, of an organism such as yourself, right, to go ahead and fight off that infection. We've effectively eradicated smallpox this way, right? So we've vaccinated people so that smallpox no longer affects humans. But we've seen, right, it's hard sometimes to go ahead and treat these viral diseases because they can rapidly burst onto the scene, right, and infect humans. And one place that we saw this was in 1918, right? There was the flu pandemic, which killed about 40 million people, right? And so there are three types of influenza, really, right? We have types A, B, and C. B and C really only affect humans, but type A also affects other organisms, and it's able to kind of make that jump, right? And it's able to make that jump. We had a massive effect, and you could see that in 1918. Within a month's time, it spreads all across the United States. And we're also seeing, right, in 2009, we saw this H1N1 pandemic, right? And so that affected, right, a tremendous number of people, right, as it jumped from avian, right? So it jumped from birds, okay? And so these are, right, causing like tremendous effects, and so showing how viruses are able to move right, between different organisms, okay? But one thing that's really interesting, and where we're going to wrap this up with, right, is that it's really estimated that 100,000 elements of the human genome come from viruses, right? And that roughly just over 8% of your DNA and my DNA is from viruses, right? And that is really kind of an astounding fact, and something that we really need to kind of like consider in terms of like how has that affected how we've changed over time. Like what have those pieces of viral DNA, right? They must have been then activated, and how are those affecting us to this day? So I hope at this point you've learned a lot about kind of the lytic cycle, the lysogenic cycle, right? How a virus operates, what sort of genetic material it's using, what they're built from, what are some common viruses, and that you kind of gained a new appreciation for how viruses work, even though they're extremely simple.